Good day. I am Brian Hoffland, President of Research to Prevent Blindness. I want to welcome you to RPB's Lunch and Learn, Eye on Dry AMD and Geographic Atrophy. Research to Prevent Blindness is the leading nonprofit organization supporting eye research directed at the prevention, treatment, or eradication of all diseases that damage and destroy sight. RPB was founded in 1960, and over our 65-year history, we have awarded more than $425 million in research grants to the most talented vision scientists at the nation's leading medical schools. As a result, RPB has been associated with nearly every single major breakthrough to the understanding and treatment of vision loss. We are incredibly proud of the breakthroughs made by RPB-supported researchers, but there's still so many treatments that need to be developed. So our work continues to fund the breakthroughs of tomorrow. In today's educational session, you will hear from two RPB-supported talented scientists about dry age-related macular degeneration and geographic atrophy, otherwise known as dry AMD and GA, and its causes, impact, and cutting-edge research. One of our speakers, Dr. James Honda, has received multiple RPB awards, including a more recent RPB Stein Innovation Award, and the other, Dr. Rinki Ratnapriya, received an RPB Career Development Award. Thank you to all the generous supporters of RPB who make our work possible. I'd especially like to thank Apellis Pharmaceuticals for their support of today's educational event. The importance of supporting medical research has never been so apparent. We thank you for your partnership to supporting innovative vision research to ensure that new breakthroughs are possible. And if you have not yet supported RPB, please consider doing so to help make sure that we're able to continue with these breakthroughs. But getting back to today's session, let me introduce our moderator, Dr. Caroline Baumel. Dr. Baumel is the leading scientist and a leading retinal specialist who has been at the forefront of retina research and patient care for over 25 years. She is the Chief Medical Officer of Apellis, as well as Professor of Ophthalmology and Director of the Retinopathy and Prematurity Service at New England Eye Center at Tufts Medical Center. She specializes in diseases and surgery of the retina and vitreous. Her interests include age-related macular degeneration, diabetic retinopathy, repair of complex retinal detachments, and retinopathy of prematurity, a childhood disease. As a board-certified ophthalmologist, she has authored over 170 publications and has been recognized with numerous prestigious awards. As our moderator, Dr. Baumel will introduce our two speakers for today and moderate the Q&A session at the end. Each speaker will speak for 20 minutes, and I will serve as timekeeper. I will give each speaker a warning at the 18-minute mark. We just want to make sure that we have time at the end for a robust Q&A. And with that, Dr. Baumel, please take it away.
Thank you. Good day, everyone. And thank you so much to Research to Prevent Blindness for organizing this important event and everything that you've done in eye care. It's really a massive effort, and so much research and critical data has been undertaken. And I want to thank these distinguished speakers for joining us today. Our discussion today will focus on dry macular degeneration, or AMD, and a specifically severe form of this disease, geographic atrophy, which is also known as GA. GA is a leading cause of vision loss in adults over 65, and this is a crucial topic because GA not only affects a large number of people, but it significantly impacts their vision and ultimately their independence. While we have multiple approved treatments for another type of macular degeneration called wet AMD, the treatment options for dry AMD, specifically geographic atrophy, remained limited until the 2023 approval of the first treatment for GA. And now we understand that among the multiple factors involved in the development of GA, abnormal complement activation, which is part of our innate immunity, plays a key role in geographic atrophy GA progression. This is what makes this an important focus for Apellis, as our treatment specifically targets within the complement system. And this research is critical for scientific advancement, innovation, but most importantly for patients who are affected with this disease. So before introducing the first speaker, I just want to remind everyone that for the Q&A session, there is an option in the toolbar at the bottom of your screen. We'll do the Q&A after both speakers have talked. So please feel free to submit your questions at any time and I'll moderate that. So let's start with Dr. Handa, who I know very well, as reputation precedes him. And I can only give you a brief tidbit of his background. He's the Robert Von Welch Professor and Chief of Retina at the Wilmer Eye Institute, Johns Hopkins University. He attended Brown and then UPenn School of Medicine, did his ophthalmology residency at Wills and his vitreoretinal surgery at Duke. He's an active vitreoretinal surgeon. He manages all types of complex retinal disorder, and his lab focuses on some of the critical events due to cigarette smoking that trigger the onset of macular degeneration. So take it away, Dr. Handa.
Thank you, Caroline. Yes, it's been it's really quite an honor to be asked to give a few words on something that I'm been passionately studying for more years than I want to admit. And I'd like to thank Brian and RPB for the multiple grants that I've been able to do. It clearly has been very instrumental in helping me forward my research. So the title of my talk is What to Know About AMD in 20 Minutes. And here are my disclosures. So really the scope of AMD, just some numbers for you to think about. Nearly 300 million people have AMD worldwide, which is almost the population of the United States. And in the United States, it's probably 3 to 5% of the population, and it's increasing because the baby boomers are growing into the age where they can get AMD. But two numbers that are really striking it, that the number of people with AMD are similar to the number of patients with invasive cancer, and it's more than double the number of patients who have Alzheimer's disease. And as a consequence, the financial burden is roughly about $5 billion in direct healthcare costs per year just in the United States. So to sort of bring it home to the patient, some studies have been done, and so people who have mild AMD has the equivalent in reduction of quality of life of someone who has heart disease, moderate angina, or has early AIDS. More moderate AMD is someone who has severe angina or someone who's chronically on hemodialysis. And someone with severe AMD is equivalent to someone who has prostate cancer with severe pain, or someone who is bedridden from a severe stroke. And there are a number of studies that show that people with AMD have the highest rates of depression relative to other adults with other eye diseases. So it clearly has a devastating impact on the patient. Next, please. So the doctor's view, really the hallmark of dry AMD are these little yellowish white spots that we call drusen. And normally there's the retinal pigment epithelium, which is a chief cell type that causes this disease, can undergo some changes that either have increased or decreased pigmentary changes that as clinicians we can see. Next slide. And here's just kind of the progression of AMD going from normal to having these drusen spots to the advanced form of geographic atrophy, the advanced dry form or the wet form. One of the interesting things is that the location of where AMD develops as well as geographic atrophy are in the same location. It's an area of the parafovea. And early in this disease, it's actually the rods, the photoreceptors are the first cells to die. And what's interesting is the fovea is very late. It's a very late event, and that's really one of the strategies behind all treatment for geographic atrophy, is really to save that central area, and that's where the complement inhibitor treatment has been beneficial. What's interesting is other conditions such as myopic degeneration. People who are very nearsighted have the disease develop in the same location. So we need to figure out what it is about this parafoveal region. And so if you take a deep dive, some prior work, the area I'm talking about is in the red box, and it's 1 to 3 millimeters off of the foveal center. The center area has the cones, which are involved in central acuity, and the rods are for adapting to light and some other senses for vision. And it's this area that is actually rod rich, but if you compare the density of the rods relative to other areas in the rods, it's actually the least dense. And so the thought is the relative lack of density in this rod-rich area of rods is what makes them vulnerable. So there could be an equal depth of rods throughout the retina, but since the density is less, this area is more vulnerable. And so we do know of risk factors of how this disease develops, certainly genetics, the number of complement genes suggesting that innate immunity or inflammation is involved. ARMS2 HTRA1 is an extracellular matrix regulator, but there are 52 variants that are associated with AMD risk. Epidemiologically, we know that there are environmental factors such as cigarette smoking, which is what I've focused most of my work. And we know that diet plays a big role, such as the Mediterranean diet, which is protective, as well as a low glycemic index diet. We also know that there are some major pathogenic pathways, going back to the epigenetics, which is sort of linking the environment with some changes that can occur to gene expression, not altering the DNA sequence, such as aging and smoking are also very, very relevant features. And so in sort of determining how AMD develops, we know the major pathogenic pathways. We know that pathologic oxidative stress, free radicals that can damage tissue, are important. We know that mitochondria, the power packs of a cell, are dysfunctional. We know that lysosomes and autophagy, the process that gets rid of damaged materials such as proteins or lipids, is also abnormal. We know that lipid abnormalities, too much fat, causes disruption. We know that the matrix, the material on which RPE cells sit, are abnormal, and that's where these drusen deposits develop. And we know that inflammation, including innate immunity of complement and the inflammasome, are key drivers in this disease. So current treatment is somewhat limited to be fully aware. I've listed the anti-complement drugs that are out there for geographic atrophy. I'm going to focus more of my talk on the earlier stages just because it dovetails with some of my research. And I'm going to talk a little bit about the treatments, the current, the AREDS, preservation vitamins that are shown in Mediterranean diet, low glycemic index diet, and link it to why it seems to work in slowing down AMD. Next slide. So if the big question is why AREDS? So AREDS are a concoction of antioxidant vitamins, and the whole intent is to reduce oxidative stress. But when you really dive down and think of why it happens, it turns out that in the central fovea, xanthophyll pigment is very, very important in protecting the macula. One of the key factors is these xanthophyll pigments are not synthesized by the body, so we need to obtain them from the diet. And so zeaxanthine and materials in the AREDS formulation have been put in to help augment the diet, and so that can certainly protect the central area. And there's recent data showing that the AREDS vitamins can actually slow the progression toward the center of the fovea. Other oxidative stress mitigation with vitamin C and vitamin E are also relevant and probably helpful in why this can slow down the progression of AMD toward advanced disease. The Mediterranean diet, which seems to be helpful in almost everything, is really a powerful deterrent or in terms of slowing down AMD toward geographic atrophy. And one of the key factors is that it eliminates the high-fat diet. And so a Mediterranean diet is rich in fish and poultry and encourages people to stay away from red meat, so that bad cholesterol is mitigated, and it's bad cholesterol that can accumulate in the macula. And I've just shown some prior work that we've done where you see cholesterol accumulating underneath the retinal pigment epithelium in the extracellular matrix we call Brooks membrane. When it gets retained, it gets oxidized, and the oxidation of these lipids can cause inflammation that then go on to develop these thick precipitates, known as basolaminar deposits, but they also can transition into drusen deposits. And a low glycemic index diet is something that people don't know as much about, but the combination of sugar and oxidative stress creates these non-enzymatic glycation reactions. It's the same reaction that causes a banana to turn brown. It's what causes one to develop a cataract. These changes are irreversible and they can damage proteins so that the function of the protein is altered, and also they're resistant to degradation. So these bad materials can accumulate within cells and disrupt the function of the cells. We know from prior work that a high glycemic index diet will induce these end-stage products called advanced glycation end products that can accumulate in the macula, and it damages the removal of these damaged molecules, so the overall function of the tissue goes down. And I put in, it's probably worthwhile for you all to Google glycemic index diet. It will surprise you. It turns out that a bagel is worse for you because more carbohydrates get absorbed into the bloodstream faster than a donut. So it will surprise you. The foundation of my work has been in smoking, and smoking is the #1 environmental risk factor for developing AMD. And to give you background, each puff of smoke has 10 to the 17th free radicals, and it has about 5,000 toxins or oxidants in smoke. So it's just a powerful chemical oxidant that can damage tissue. Our work has really shown that the mitochondria, or the power packs of the cell, really can accelerate aging and cell death and cause dysfunction of the retinal pigment epithelium that can then damage photoreceptors, which are the cells that we see with. And it also can damage lysosomes as well. And so one of the things, this is an example of RPE cells from our animal model that we expose mice to smoke. And you can see that on the left panel, where mice are raised in air, there's this hexagonal normal appearance to the retinal pigment epithelium. But when we put them in smoke, there's a degenerative process. You can see the elongated, enlarged, misshapen RPE cells. And these cells are not able to take care of the retina as well. And so vision can precipitously drop. So I'm going to pivot to talking a little bit about some of the work that we're doing on the Stein Innovation Award, which I feel very fortunate to begin with. So we've sort of extended our work thinking about, well, asking the question, what are the consequences of mitochondrial injury to the RPE, and are there signals that spread to other cells that can damage ultimately photoreceptors? And so one of the consequences of mitochondrial dysfunction is that we became interested in tRNAs. So tRNAs are nucleic acids that actually bind an amino acid, and they also bind to the mRNA template. So it actually helps the growing peptide into a protein. So it helps produce the protein. Stress or oxidative stress, including from smoking, can cause these tRNAs to fragment, and they have always been thought to be waste until recently, when there are some studies that suggest that they can have biological activity. And nobody has really studied whether these tRNAs that are made only in the mitochondria have any role or any function. So that's what we set out to do. And mitochondria are interesting. They're kind of like bacteria that got stuck into cells at some point, and they have their kind of own system where they make some of their own proteins. And so we wanted to look specifically there. We poisoned RPE cells with rotenone to damage the mitochondria, and then we looked at tRNA fragments. If you look at the next slide, we'll show the distribution of these tRNA fragments that occur in the mitochondria or in the cell cytoplasm. And there's a bias toward mitochondrial tRNA fragments increasing in the mitochondria, whereas the tRNA fragments from the cell or in the cytoplasm seem to decrease in their abundance. Next slide. So we focused our work on one particular mitochondrial tRNA fragment we call the GLIN-TTG. We did some basic tests where we would add these synthesized mitochondrial tRNA fragments to our RPE cells, and we got them to be absorbed into mitochondria, and we looked at the mitochondrial function. So TMRM is a way to look at the electrical charge of mitochondria that needs to have this charge in order to produce ATP. That's impaired with these fragments. ATP goes down. MitoTracker is a way to look at the viable or healthy mitochondria. That goes down. And MitoSox, which is a measure of looking at the free radicals or the oxidative stress that's generated, since mitochondria generate a lot, that goes way up. So it certainly shows that mitochondrial function is impaired. And when we looked a little bit more, asked specifically why, the electron transport complex is an area that's involved in making the ATP molecules. And when we added these mito-tRNA fragments, it decreased the complex production, but also importantly, the activity. And so that stalls and prevents ATP from being made. Next slide. And part of the reason we think that happens is when we looked at the structure of these tRNA fragments, they look like they have the ability to bind to the mitochondrial DNA. And we theorized that when they bind to the DNA, they would inhibit the gene expression of mitochondrial-derived genes. And indeed, when we added these tRNA fragments, they inhibited a number of the genes that are produced by the mitochondrial-derived DNA. So it looks like the decreased expression of these genes is what is responsible for impairing the overall mitochondrial function. So we wanted to find out if, since these fragments are being produced by the RPE cells, can they spread disease? In other words, and the question is how, and we've been interested in extracellular vesicles, or these membranous vesicles that can take on cellular contents, get released by cells and spread throughout the body. We're interested because these vesicles are, they will protect the contents inside of them. And indeed, we know that when we stress the cells, we can uncover these vesicles. And here's some images, electron microscopic images of the little vesicles that are released from RPE cells after we add cigarette smoke to them. When we looked at the contents of them, they did indeed contain high concentrations of these mitochondrial tRNA fragments. So we then asked the question if we could add these mitochondrial tRNA fragments to other cell types and see if it affects it, both whether they're in these EVs or whether they're naked. And indeed, when we grew iPS-derived stem cells into retinal organoids, with collaboration with my colleague Don Zach, these took about 200 days to grow from stem cells into retinas, which do have photoreceptors. And so we added the tRNA fragments to these photoreceptors to see if the mitochondrial function in these retinas would be impaired. And the way this assay works is that the green dye increases when the mitochondria get damaged, and the red dye goes away. And you can see the vehicle, everything looks normal, but you can see the red increases, the red decreases and the green increases. And on the chart, you can see the drop from adding the tRNA fragments. So it certainly suggests that when we add the tRNA fragments to retina, they're able to damage the photoreceptors of these retinal organoids. So that's kind of a summary. I just wanted to end.
At the 18-minute mark, you should wrap up in the next 2 minutes, please.
Great. So I just wanted to end by saying that as I've thought about this disease, and I'm sort of obsessed with this disease, is really, in order to come up with really effective treatment, we need to answer these 4 simple questions. How do the genetic variants cause geographic atrophy or AMD? We still really don't know. What are the major GA causing pathways? Well, I listed some, but the problem is we don't really know the order, the rank order. So is complement the most important? Is mitochondrial dysfunction the most important? We actually don't know. And if we could prioritize them, that might help us to hone in on what specific treatment is needed, whether even a single treatment is needed. And the last question is really, do these pathways interact with one another? That's relevant, like chemotherapy, where we need multiple drugs to have effective treatment. currently we're all on monotherapy. But if we know that these pathologic pathways intersect with one another, we might be able to find the branch point where we can have one drug treat multiple pathways, or we may be forced to have multiple pathways because they don't intersect with one another. So I think these are really critical questions to answer. So I just wanted to end with hope you can appreciate the complexities of AMD. I tried to give a presentation from the patient's version, what the doctor sees, some current treatment, why it works, and some of the recent work we've looked at to understand how the disease spreads. And again, to end with some of the key questions that I think we as a group, whether we're in academia or in industry, need to address to get more effective treatment than we have currently. And I will end it there. Thank you.
Thank you. That was such a great talk. I have a lot of questions, so hopefully we'll have some time at the end. Now I'm really happy to introduce our second speaker, Dr. Rinki Ratnapriya. And just very brief summary, because I want to give her more time to talk. She is a PhD. She's an assistant professor from Department of Ophthalmology, Baylor College of Medicine in Texas. She earned her BSc at Delhi University, and then she followed that with a PhD in human genetics. She completed her postdoc fellowship in ocular genomics at the National Eye Institute. And I'm just going to wrap it up by saying her research focuses on elucidating disease pathogenesis using very large genomic, transcriptomic, and epigenetic data sets. And really, this is the sort of thing we need to understand this very, very complex disease pathogenesis that affects so many of our patients. So she's going to tell us how it's related to retina disease. Thank you.
Thank you, Caroline. That was such a nice introduction. And so let's get started. So I have no disclosures. And I would like to start by thanking RPB for inviting me to give this webinar, as well as giving me the Career Development Award, which was really instrumental in starting my career when I was just out of my postdoc. So thank you. So very briefly, you guys heard from Dr. Handa that AMD is a very important disease, leading cause of incurable vision loss in elderly worldwide, and the dysfunction of and the death of the photoreceptor support system, which includes RPE, retina, and the choroid, is the primary cause of vision loss, and it is a progressive disease with the early, intermediate, and late stages of the disease, where the late stage where we are focusing today on the late AMD, dry, and the wet. So since I work on the genetics, let me just lay down the, why it's so important, right? So we heard that there are 52 genetic variants that are associated with the disease. So yes, but how does it help the patients, right? So we know that 90% of the drugs that enter the clinical trial, they fail to reach the sufficient efficacy or the safety, and that's why it's a very expensive business to make drugs. So around 10, 12 years ago, it was demonstrated that the drug targets that are based on human genetic diseases or the robust clinical biomarkers, they are more likely to succeed. As you can see here, this is the odds ratio, and if you have a gene that is coming from OMIM gene is like a database for the human disease genes, or from the GWAS, their odds ratio is clearly much higher, so they are more likely to succeed. And that led to substantial investment in human genetic and genomics for the potential drug targets. And in the last few years, you would have seen many companies also starting to investigate the human genetics to mine these targets. As shown here, so this progress has been really very good for the rare diseases, which is shown in the orange bar, which makes sense because in the rare disease, when you have a mutation, you have a clear idea about what is the gene involved. However, that enthusiasm is less for the common diseases because of some of the reasons that I'm going to show you in the further slides. So we know that GWAS is important, but for common diseases or complex diseases, it has not been very successful. Why? So as we know that AMD is a complex disease. So what is a complex disease? So it means that it is caused by a combination of different factors. So the genetics play an important role. We know that in the last big consortium paper in 2024, it has been 63 loci and the multiple genes that have been identified, as well as its environmental factor. We just heard about the smoking, as well as the aging is the biggest risk, non-genetic risk factor for AMD, as well as the AMD heterogeneity and severity that can play a role in disease outcome. So how do we identify the genetic markers in a complex disease like AMD? So the very typical method that is used is called the genome-wide association study. So what is it? It's just very simple premise that if the disease is common, the variants or the gene associated with the disease will also be common, what we call as the common disease, common variant hypothesis. So we just collect a bunch of cases and bunch of controls, and then when we are genotyping, we are looking for the variants that have higher or lower frequency in cases than controls. So if the frequency is higher, then you would call that as a risk variant. And if the frequency is lower, then it would be a protective variant. So that's why, see, here we are using the term variants. We are saying risk variants. We are not calling mutations as you would use for a common disease like inherited retinal diseases. And what we see then is that classic Manhattan plot, which looks like a Manhattan, line. So everything which is reaching that statistical significance is shown above this dotted line. And those are the locations. So all we know is that those are locations we should be focusing on. So AMD has been really a poster child for GWAS successes. Right? Why I say that, the first ever GWAS was performed in AMD with merely 96 cases and 50 controls that led to the CFH identification. Followed by in 2016, a large GWAS with 16,000 cases and 17,000 controls led to identification of 34 loci. And recently, this number has increased to 63 associated loci. So where does it stand right now, the genetic architecture of AMD? So we have two clear distinct categories. One is the common allele with the high effect. However, this is more like an exception than rule. We have only CFH and ARMS2, which has a very high odds ratio. Most of the GWAS variants, not in AMD, even beyond AMD, they are common allele with moderate to low effect. And then in rare instances, we will also have the rare allele with high effect, and we know the examples of the rare variant in CFH, CFI, and C3 have demonstrated. So what that helps, it immediately points out to the gene that we should be focusing on. So the key questions in GWAS, right? So I told you that when you do the GWAS, you identify risk variant. So roughly you have a region in the genome that you can focus on. That is still very, very important because you could just exclude 99% of the genome and you could focus on a very small genomic search area. However, majority of the GWAS variants are non-coding, and we don't understand it. Over the last couple of years, we have been getting more information that earlier the whole non-coding genome was called the junk genome, right? It was not long time ago that we would read that in even textbooks. But we are realizing that they're a very important part of the genome, and but when we identify these risk variants that are non-coding, then we don't know what is the causal variant. So just because it's associated, we never call that causal. We call that it is associated. So when you have this variant, you are highly likely to have. Now, the second problem is that before this, we have something called linkage disequilibrium in human genome. So what it simply means that the variants travel together. We have blocks of the DNA. So that's why in that region, there'll be several. So there will be one with the lowest p-value. However, there'll be many, many variants that are equally strongly associated with the phenotype. However, we would not know which exactly is the causal variant. So that has been one of the key challenge when we have the GWAS, but we can't immediately reach out to which is the gene or which is the pathway. Then the disease relevant cell type, all you get is the genomic region. We need to figure out it is working through RPE, it is working through retina or choroid, or it is on one side one gene, the second side it's a different gene, or it's a cell type specific regulation, or this regulation is shared. And finally, connecting to the known biology has been difficult, because if you will look at the GWAS region, you will have some are very clear, like, with CFH. First, we had a very high odds ratio, then we had the rare variants identified. So it was very clearly it was easy to kind of find out that, okay, CFH is the target gene. But for majority of the loci, we don't have that kind of information. So we don't know what are the target genes and the risk genes, how they interact and what pathways they are involved in. It is not very clear, because in one locus, you might have a dozen gene that is encompassing that, and it is hard to pinpoint that which is the target gene responsible for AMD. So how do we comprehend the molecular mapping of the GWAS variant? So I told you that majority of the GWAS variants, not only in AMD, but across all GWASs, they are located in the non-coding genome. And in 2012, it was shown that these variants, they often overlap with the enhancers, promoters, and the open chromatin regions. And that would go on to say that they are most likely to exert their effect through gene expression regulation. So they are sitting near the gene, and then they are, they are affecting the gene expression regulation. So what we call as the EQTL mapping. So what it simply is that there is a SNP, which is located here, and there are two genes, gene A and gene B. So if you have the expression profile, which is the RNA, and genotyping, which is the SNP profile, which is the genotyping data. And if you connected that, so how to understand if this is the variant and there are the two genes, you can see that with respect to, we are all diploid. So there could be, like, if the variant is A to T, we will have 3 AA, AT, or TT. However, you can see the gene expression, if you measure gene A and gene B, the light green, which is gene A, expression is decreasing with respect to different genotypes, whereas the gene B remains similar. So what you could conclude from here is that the gene, this particular variant is regulating the gene expression. That's what we call an EQTL mapping. So during my postdoc, I had an opportunity to work with generate the largest EQTL map for the retina, where we took over 500 human donor retina and then collected their RNA and DNA and generated a EQTL map. So what we found was there were around 14,000 EQTLs that were regulating 10,000 genes. So in retina, we identified around like 16,000 or 18,000 genes to be expressed. And around 10,000 of them, they were under regulation by some genetic variant. And when we compare to around the same time, GTEx, which is another big, large data set where they are looking at the EQTL, basically genetic variant-led regulation across different tissues in the body, when we compare our data to that, we realize that a large proportion of the EQTLs were shared, but retina also had some tissue-specific UTL, which will be same for every tissue that you would be looking at. So a lot of the regulations are shared, which again makes sense that if you take two tissues, we know that some of the genes will be equally expressed, but some of them will have the tissue-specific function, like rhodopsin in retina. So going forward, when we started look at, we see this disease heterogeneity. So in AMD patients, we don't really say class A or class B. So even though we had like over 100 samples, we could not, when we performed the differential expression, we were not finding many genes. So what we see more like this. So we have like a lot of, there's a little bit of mixing up, some of the normal looking more like controls as a disease and vice versa. So this becomes a very typical machine learning problem. So when I started my lab, we developed this machine learning approach to identify AMD genes. So what you take is that you take all of the genes that are expressed in the human cases and controls of the retina, and then you go through different iteration of selecting the genes with the highest variance. And then we came up with the 81 gene that were uh, put in the machine learning model. And what we identified, we tried like 4 different machine learning algorithms, and we found that the mean AUC is 0.8. So in simple terms, what it says is that we were able to discriminate between the AMD and non-AMD with high accuracy. So it was very exciting to see these 81 genes because many of them were novel candidates. So we wanted to know what are these genes involved in, so we performed a gene co-expression network analysis, which is nothing but learning about the connectivity between the genes at the transcriptional level. So what we found was the immune response, ECM, and the complement, which we know are very important for AMD. These genes were enriched in that. And between the control and the AMD, we were seeing some differences between these connectivity. And more specifically, we found that the complement pathway module that was not conserved between the disease and the non-disease, which again made good sense with what we know about the biology of AMD. So since in this set, we did not have many of the known AMD genes, so we were like, okay, so how do we know that they are important for AMD? So what we did, like we looked at specifically their relationship with the known AMD genes by correlation, and what you're looking at is a heat map. And what I would like you to focus on, you can see this is the control heat map and this is the late heat map. And in the middle, you're seeing wherever you see this yellow box, that means the difference between the correlation between these 81 genes and the known genes are significant. So you see a lot of green, yellow boxes. That means that even though the gene is expressed in both cases and controls, they are more correlated, they are more functionally working together in the disease compared to the control. That again gave us a good confidence that these are the genes that could be important for AMD processes. So where are they expressed? We wanted to know because retina, all of this work was done in the bulk tissue, and we know that retina is made out of many different kind of cell types. So we use the single cell RNA-seq data, and we see that the 81 genes are largely expressed in retinal immune cells, especially the microglia and astrocyte and the Muller glia. And we know that immune dysregulation and the complement pathways are important for AMD. So again, going by that where these genes we are seeing expression made good sense with the AMD biology. So we went back to our original human data, which was the bulk data, and we tried to call what I would say a digital deconvolution. So basically, bulk RNA sequence, you can think of each of these Lego pieces of the different cell types, and you can deconvolute. So you can just separate them by colors and then kind of get the information for each one of the cell types. So we utilize multiple different methods, and what we find is that the microglia, astrocyte, and RGC, they were different between normal and the late. And especially microglia, you can see that it was different across all different stages of starting from early, intermediate, and late. So finally we went, so this is the one data set in human, since we are very, very heterogeneous, it is important to have a different data set, like a different, what you can call as a validation set. So we took the single cell between the cases and controls, and we could replicate a lot of our findings. So then we also went back to the GWAS data, and we found, so why we are going back to the GWAS data, because going back to the first thing that we were talking about, if it has a genetic basis, it has more likely to succeed. So what if any of these 81 genes has a genetic basis? So we went back to the GWAS data, and indeed we see something which is a suggestive association. So GWAS is a statistical game, right? When you were using 50 samples and 96 controls, you were identifying only CFH. When you're using 16,000 cases and 18,000 controls, you are identifying 34 loci. So there are still more loci to be discovered, and it needs more sample size. So this is like also a kind of alternate way. You could look at the suggestive association and use the data from the transcriptome side and identify a new gene. So we focused on this one locus where we were able to identify a variant, and we showed also that in the luciferase, that particular segment of the genome, non-coding genome, has a enhancer activity as shown in these luciferase assay.
Dr. Ratnapriya, you should wrap up in the next 2 minutes, please.
Okay. So then I will just try to kind of, so we started with the DNA. Now I showed you the data of the RNA. So the next part is really looking at the chromatin, so epigenome. So what we are doing now is really looking at all the ATAC-seq and the methylation data of the retina and the RPE. And what we have found, we generated the profiles, and we see there is a good concordance, what you would expect. And more specifically, when we integrate the AMD GWAS data to identify what cell types are important, and you can see that, again, both RPE and the retina is highlighted. However, RPE has higher heritability. So that means, in simple term, you can think of it that it is it's contributing more to the disease. However, retina is made of different cell types. So among the cell types, we see the microglia and astrocyte, which is very similar to what we had seen in the transcriptomic analysis. So in the interest of time, I will just move on from this data, and I will conclude that integration of the functional genomics and the GWAS can identify a disease-relevant cell type. We can actually try to understand the biology underlying AMD. Functional genomics and machine learning can identify gene signatures, and we can also use these integrative approaches to offer valuable insights into the disease mechanism and potential therapeutic targets. So with that, I would like to thank, and I'll take any questions you might have.
Well, thank you. Thank you for these great talks. And, I think these talks really highlight the complexity of this disease. We have two amazing, researchers who have been dedicating their careers to this disease, both studying different facets of this disease. And I'm really interested how they would come together. One, Dr. Hannes, focusing on a big risk factor, smoking, and how it affects mitochondria, which could actually relate to many other diseases. And Dr. Ratnapriya focusing on the wider view, which will give us a lot of information. There's one question, there's one or two questions here. One is to Dr. Rinke. I hope you don't mind me calling it. Everyone calls you Rinke.
Yeah, that's easier, yes.
What do you think, when someone asked, do you anticipate development of a polygenic risk score for AMD? But I'm even going to simplify that more. Patients come in to see me, and I would love to give them a genetic test where I could say, if you smoke, your risk of AMD is going to be higher because you have this disease. Do you anticipate some test, but something we can do that would be also simple for patients? And how far off are we?
Yes, I think that's definitely possible. However, with the current, so as I showed in my, presentation, when we do the GWAS, we have a good idea where to look, but we don't have a very good idea what to look at, right? So, and that's till we kind of fill that gap, and that's at least a tiny part that we are trying to do is identify the functional variant within those loci. So that would be the first. The second thing is, of course, the environmental factor. That's Dr. Handa is showing very beautifully that how much impact of those this can have on. So in human, we are also very complex, right? To quantifying this, especially the environmental part. I think the genetics would be easier once we have a good handle on what is the causal variant, that you can say that, yes, definitely we can generate the PRS. So currently the PRS you can generate, however, it's sometimes it's possible that since it is not built on the actual causal variant, it is built on the risk variant, it is not very accurate. Whereas if we try to kind of fill that gap from the risk variant to the causal variant and then build the PRS at the genetic based PRS, probably would be much likely to come sooner. Environmental one, I would say it's kind of till we get a good handle on how to quantitate, because these are all numbers, right? You need to put in some kind of, this one has this much of burden, this particular aspect has this much of burden. That's how you build the risk score. So, but yes, definitely I feel that, it can be built.
So what do you say about the genetic tests that come with, there's some commercial genetic tests that, you might get for Christmas under your tree. Someone gives you one of these tests. And I know they do have a macular degeneration. One of them has a macular degeneration risk. Sometimes patients come to me worried about that. What do you feel about testing, and what do you suggest we tell patients about that and the community?
Yeah, so I think it has to be taken with a bit of caution, right? I mean, it's like it is kind of also very fashionable, right? The genomics became so available, it became so much cheaper to do it, and then everybody is doing. But you have to realize for AMD especially, because we have some very, as I was showing, the CFH and ARMS2, they are really the exceptions in the GWAS, not really the rule, where we have such strong odds ratio associated with the disease. So if you somebody has those variants, their chances of developing like homozygous variant, risk variant, their chances of developing AMD is much, much higher. But if you are talking about major, those are just the two we are talking about. There are so many 32 other, not 61 other loci. And there the odds ratio is much lower. So just because you have the genetic variant, it does not mean that, and it's one of the genetic variant, right? It's a combination. It's first of all, you don't need to have all the 63 risk variant to develop the disease. Plus, even if you have all of the, a lot of those risk variant, if you do not have the right environment, you may not develop the disease. So right now, I think those genetic tests probably are not very useful. That's what I would conclude.
Great. Dr. Handa, you do a lot of work on smoking. Mitochondria is a very hot topic, and I really liked seeing how you had your models in the lab are kind of approximating retina tissue in the real world. What do you tell patients that are smokers about their risk for eye disease? And do you think there's anything that we could be doing better as a profession about smoking and blindness?
Yeah, it's a powerful. I mean, I clearly tell my patients that they're at substantially increased risk. The real challenge comes with smoking cessation. So it's a very difficult thing to stop. I don't know of any great programs that can get somebody to stop smoking, as opposed to there's a pretty substantial literature showing how diet alteration can be effective. So I think we need to learn how modulating diet can be applied to smoking. Certainly, if you look at the overall adverse impact on smoking throughout the body, I think there can be some powerful public health messages that can be sent that were at one point, and smoking has decreased slowly, but just not enough at this point.
Mm-hmm. What, if you have one key message, Dr. Handa, from your talk today, one key takeaway for the science and maybe one for the public, what would that be?
So I think overall from the science, I think if we were able to prioritize the pathogenic signals, I think that would be probably a quicker pathway to get more effective treatments. I think coming up with a target that has multiple pathogenic pathways is more complicated. So I think that's the most powerful message. We need to really understand, really prioritize these pathogenic signals. I'm sorry, and so the second one was the message for our patients?
What would you tell doctors or patients about this? Because, we do have now some treatments for geographic atrophy. It's the first time we've been able to slow the growth of geographic atrophy, but this is it's complicated disease to study because we don't notice the vision improvements necessarily, or we've tested patients too far along in the disease course. So what are you going to tell your patients that come in that, are losing vision from macular degeneration? We have treatment for wet macular degeneration. It's not perfect all the time for every patient either. What would you tell them?
Yeah, I mean,
I'm asking you for a bionic eye. You probably get that as well.
Yeah. Well, I think it's two-sided. One is, we go through a very careful discussion on the pros and the cons of the current anti-complement treatments out there. We talk about diet control, and obviously controlling blood pressure, cholesterol, things like that. And then after a deep discussion of the pros and the cons, I leave it to the patient to decide, and in part because we do know that all these treatments, the vision isn't going to improve, but we're trying to prevent the vision from dropping off the cliff. And so we leave a lot of it to our patients to make that ultimate decision for the current treatments. And then I like to provide a little bit of hope, because I think there's a lot of really very interesting research and advancements from the clinical trials for RPE transplantation, which are several both in the private sector as well as at the NIH, and other potential targets and greater insight into our understanding, and the acceleration of our understanding. If you look at what we know today versus even 5 years ago, I think it's substantially different. So I do provide them with some hope as well.
Great. And then Dr. Rinke, why don't you give us the key message you want everyone to take away from your talk today?
Well, I would say that genetics is very powerful. That's like one thing. So even though we don't have all the tools to understand, what we know about the AMD biology, but a lot of work is being done in this area. And in coming years, I anticipate that we will have much better understanding of the biology, at least that is underlying the genetic basis of AMD. And that would kind of, again, provide, as Dr. Handa was saying, your targets, and that could be therapeutically important for patients. So,
well, thank you so much for your time today, and thank you to everyone for joining in and for the questions. I'll pass it back to Brian.
Well, thank you very much. I wanted to thank you, Dr. Baumel, for a very able moderation of today's session, today's event. And I also want to thank Dr. Handa for what I thought was just an excellent overview of AMD that is, we should model it and send it to every patient, or prospective patient. And also giving us sort of an overview and a glimpse of the importance of the possible importance of mitochondrial dysfunction in the development of AMD. And Dr. Ratnaprema, rather, I want to thank you for really talking about the importance of the genetics perspective. And my goodness, 81 different genetic variants, that it really shows the complexity of this disease. But I think it holds out hope that we can develop robust clinical markers, and that down the road it may be possible to really identify those patients that will benefit the most from these new treatments coming on the market. And I think that'll be a very great interest and importance to those who pay for the treatment as well, both public payers and insurers and private patients. But thank you all. And I want to thank everyone who tuned into the session. The recording of this Lunch and Learn will be made available to everyone who participated. We really encourage you to disseminate it to your networks. Really, I think the important part of the dissemination comes after the event itself, when it's shared amongst people. I know we get many hits on our YouTube channel, but we encourage you to disseminate it to your audience as well, both at Baylor and at Johns Hopkins and at Apellis. So again, thank you to all, and it's been a great session. I appreciate the participation of everyone. Thank you.